And we're recording, so welcome. We are ready to talk about chapter 29. After a long 2 semesters, the last chapter in the Anatomy and Physiology textbook. And that chapter begins with a consideration of 2 terms related to our reproduction: development and inheritance. Now, when I hear the word development in biology, I always think of the same thing, and that is, given that our body is made of trillions of cells, you might think there'd be some variety with how we reproduce, but no, we go back to the 1-cell stage. Fertilization of a sperm on an egg produces a single fertilized egg, and that's how our life starts. Mitosis is going to produce more cells, and cell differentiation is going to specialize those cells to different functions. Those different specializations are what we call tissue. Do they form surfaces? Do they support and connect? Do they produce movement? Do they produce signals? Those are the basic 4 types that result from differentiation. Now, studying the process from conception, that process of fertilization, all the way to death, and the different typical stages and the different typical changes from one stage to another, that's what we mean by development. So the first part of development, we'll be getting that single fertilized egg to a multicellular stage that we call an embryo, and then on to a fetus, and basically produce increases in size and differences in tissues until we have a fetus that is ready for live birth. And that's what we'll kind of focus on during the first part of this lecture. The second term is inheritance. What are those principles that govern the transfer of genetic material from generation to generation? What am I talking about? I'm talking about what is my contribution to my offspring. What are the rules that govern how I can affect the genetic characteristics of my offspring? We see the power of genetics in producing identical structures when we witness the phenomenon of twins, but not just any twin, monozygotic twins. That means one egg and one sperm produce a cell which divides to form the 2-cell stage, but those cells, instead of staying together and producing one offspring, somehow get separated, and they implant separately. Now, when those monozygotic twins emerge and are born, they're healthy, their structural, their physical similarity is striking. You look at them, they look alike. So it shows that genetics has power to produce identical structures. The fact that except for monozygotic twins, you see differences between any pair of individuals in the human population speaks to the real role of sex and of inheritance. And that is to produce variety. We not only don't produce identical offspring, we can't, because we're limited to sexual reproduction. Its whole point is to produce diversity in offspring in the population. So looking at these 2 topics through the chapter, we'll review this idea of differentiation, which is basically a generalized cell or a stem cell becoming a cell that has, well, you might think you might say it has its education. It's been trained to its job. So after that change from a stem cell to a specialized cell, it will perform one kind of task for the rest of its life. This is due to differential gene expression from genes in the nucleus. Fertilization or conception, we've already explained, and that's really when development begins, from that stage of the single squishy cell. Now I'm happy, at last, a figure that accurately represents the size of the egg compared to the size of the sperm. Not in any other figure up to this point have we achieved that. Here we have the egg. Now, given the photography of this, it is so good, I rather suspect this is from an in vitro fertilization. where a harvested egg in a petri dish is being introduced to sperm to achieve the fertilized egg, but the photograph clearly shows this single cell that the female body produces. What a huge cell, full of all the organelles you'll need, all the resources, the stored carbon fragments that'll be used to make ATP, everything you need to keep a cell well supplied. And then take this individual right here, here's an individual head of a sperm. So this is more like a basketball compared to a BB. It does have this long, extended, very thin, rope-like flagella that provides its propulsion. But in fact, the relative size is witness itself. This is only a half nucleus with a motor on it. Even with this aggregation of sperm right at the outer covering of the egg, when they arrive, one sperm will penetrate the membrane and it causes an instantaneous change to the membrane. So one and only one sperm, based on biochemical regulation, is going to achieve fertilization. Once that fertilization occurs, cell division is going to begin very rapidly and in a very different and unique way. Normally, when you divide a cell into two daughter cells, there's then a pause while these two daughter cells grow back to the mature size, and then they're ready to divide again. In the fertilized egg, you get first cleavage. These are basically daughter cells that are half the size, and then you almost immediately, all you need to do is take time to replicate the DNA in the chromosomes, and you divide again. You can see these are about quarter-sized. And if you divide another one, it'll be about 1/8 and then 1/16. What you do is you take the available resources of the cytoplasm and you just divide as quickly as you can. This also illustrates something about fertilization. Ideal timing would be the delivery of the sperm down here in the vagina in time for them to swim all the way up through the cervix, through the uterus, out this uterine tube, basically be waiting in this vicinity when the ovulation occurs. The sooner the fertilization occurs, the better. So you can see 1, 2 divisions in the first 2 days as this non-motile fertilized egg is bouncing down the uterine tube. Day 3, day 4, you're achieving a large enough number of cells that you no longer can be sure of their number. I mean, you can see 7 cells there, but you kind of can't tell about the packing on the other side. This is called the solid ball stage, or the morula, early and advanced. These divisions continue. And you notice there's not a really much of an increase in size yet. You're basically just dividing up. Copying the nucleus, dividing the cytoplasm. Copying the nucleus, dividing the cytoplasm. But pretty soon, you get to a stage where if that remains a solid ball, there are too many cells between you and the outside fluid. Diffusion isn't going to bring in the oxygen and the nutrients that you need and get rid of the waste. So at that point, changes begin to appear. And you have a stage called the blastula, followed by a stage called the gastrula. Here, you'll see folding, and you'll see migration of cells to form a ball of different appearance. Now, when you make a hollow ball with a hole in the middle, then you're going to see some increase in size. This blastocele becomes a part of the body cavity, and this inner cell mass will become part of that we will call the embryo, the actual offspring. Days 7 to 10 is witnessing this stage or a later stage contacting the endometrium here in the uterus. And there's an instant reaction as the blastocyst grows into the endometrium and the endometrium grows out to surround the base of the blastocyst. And you have a process called implantation. This is the approach. It's occurring here in the wall of the uterus somewhere. You can see this approach, the uterine glands that are keeping this moist and full of the right fluids. And the blastocele approaches and contacts and immediately begins proliferating. Basically, this tropoblast and these developing strands called villi are anchoring the blastocyst to the lining of the uterus, the endometrium. We see the development of an amniotic cavity. The amnion is a special fluid and a fluid-filled sac within our embryos that will surround and protect the embryo as it develops. As we go a few more days, day 9 to day 14, you can see a surrounding. Now it's almost like the blastocyst burrows in at the same time that the endometrium grows out to surround it. Pretty soon you have this enclosed and protected embryo. Here is the amnion and a yolk sac, just in all other eggs. A yolk that is stored nutrients that are supporting the development of these cells in this region, surrounding mesoderm and other areas of the embryo. Now, at this point, about week 2, it's no longer simply a ball of cells. You know, a ball is pretty much featureless from any direction. It looks round, but you can clearly see a kind of a elongation here in the up and down direction and a clear fold running the length of the embryo, basically forming this little T here around a single opening. Up at the other end, you see these two flaps paired left and right. This defines the embryo in this stage of development. Now, this is a monkey embryo, but only because we recorded that do we really know the difference. Most vertebrate embryos this far basically go through these same stages. What we're seeing here is the future head and a neural plate. The spinal cord axis will develop along this line, and these individual segments are called somites. Somites are repeated blocks that run parallel to each other on either side of this axis. And they are the precursors of the vertebral division of the spinal column. Neural folds are seen here. Down here, the future tail of the embryo. And as I said, if you're talking about a vertebrate, not even a mammal, but a vertebrate, so you compare the embryo of a human with that of a, let's say, a lion or a cow, but not even just mammals. If you compare it to a chicken, if you compare it to a snake, compare it to a bat, animals with very, very different bodily organization as adults. To this stage, they look very, very similar. So here we are at a month, and we can clearly see this enclosed developing tissue with this swoop, this kind of fishhook looking appearance, blue with segments, a tail fold and an embryonic head fold attached to this yolk sac and body stalk within the amniotic fluid. We basically will see the surrounding endometrium now completely enclosing, and the development in that surrounding uterine tissue of the beginnings of the placental attachment. The same rules apply. Early on, when this is a long, narrow strand, then diffusion can get things in and out. But as the cells become more numerous and they pile on top of each other, you need some specialized system for bringing in resources and removing wastes. And that's what the placenta will do. Here is that embryo, and as I said, no matter what you start with, if it's a vertebrate, it's going to look about like this at a month. Notice the segmental development that's clear here along the back, those somites beginning to form the vertebral division. You can see the tip of the shoulder here and the arm bud beginning to develop, and the tip of the leg, and this here would be the hip. So what does that mean? That means this long extension right here is a long, beautiful tail that develops in all vertebrate embryos, including humans. We can see the heart imaged here as two distinct chambers, upper and lower, because the heart develops quickly and begins moving blood around the inside of this developing embryo. We also see pharyngeal arches. Now, in fish, that becomes gills. And in amphibians, it becomes gills at birth, which then later change to internal lungs. We see a primordial eye spot and the development of an ear. Later on, we see this picture for the uterus as a whole with a clear placental development. Now, this placenta is interesting because the orange part inside, including this embryo, the amniotic sac, amniotic fluid, all of that is coming from the fertilized egg. And the fertilized egg side has a kind of a capillary network here on its side that can pick up resources from that purple area that's common. The placenta itself is delivering resources, oxygen and nutrients, to that exchange area. But it's important to realize blood from the mother does not flow in to the embryo at any time during its development. That exchange occurs in a kind of an open area between capillaries. This is 2 months, clearly a more human appearance with a gigantic head compared to the size of the body and limbs. You can see toes. You can see the remnants of a little tail as the body gets large enough that it grows beyond it. Basically just absorbs it. The tail does not extend beyond the hip girdle at this point in development. But clearly you can see the beginning of human features and anatomy. A little later you can see the umbilical cord clearly allowing this fetus to move within the amniotic fluid, and that fluid is providing a hydraulic cushioning. So any impact through the body wall, it's basically going to be transferred through the solid tissue, but here it's basically just going to produce waves. So it will move around, but it won't be sharply impacted by casual forces. Week 12, 3 months into the first trimester, and definitely more human appearance. This is a good picture showing you the development and separation of the fingers. The eyes are developing. The nearly transparent skull is showing you the development and the division of the frontal lobes of the brain. All within this amniotic sac, the attachment of the umbilical cord to the placenta shown here. Second trimester is just fast growth, more mitosis, more differentiation, and a growing, at first in concert with and then later outgrowing the growth of the surrounding placenta. The third trimester, most of the organ systems become ready. Notice that word ready. The only organ that's really functioning is the heart. It is moving blood around the infant's, around the embryo, the fetus's body. The growth rate starts to slow at the end of the third trimester, and this is when there's the largest weight gain with respect to ounces. The enlarged uterus and the fetus itself will press outward in all directions. Kind of shows you the reason for the soft wall of the abdomen so that it can expand out. It will compress the organs and basically place a great deal of pressure on them. So here's 4 months early in the trimester. At this point, you can see individual fingers and knuckle creases and fingernails. This is an actual endoscope visualizing within a mother's womb. The positioning is still low at this point in the pelvic girdle. You can see it surrounded by important and strong bone structure. This is a time when it's beginning to show. And at later development, pregnancy at full term, this soft wall is the abdomen. On the anterior and lateral aspects are the only thing that can push out and expand. You can't push the spine, although there is tremendous pressure on the spine because it's having to support weight that is increasingly moving out and away from this axis of bony support. So lower back pain is a problem as the fetus develops. But the fetus isn't the entire weight, of course. There is the water in the surrounding amniotic sac. There is also the extensive placenta and the growth of the uterus itself that contribute to the total weight gain of the mother. So from this position in the uterus, we see a number of factors that are coming to bear. So what is it that actually signals the time for parturition, for delivery? In the placenta, we see estrogens increasing, and the smooth muscle cells of the muscle layer begin to contract, initially at low intensity and far apart. As the weight increases, as the uterus stretches, estrogens increase further, and that increase is going to increase the sensitivity of the smooth muscle fibers to a hormone from the hypothalamus called oxytocin. Relaxin is produced by the placenta, which begins to soften the pelvic articulations. Now that means especially the pelvic pubic symphysis begins to stretch a little, giving more room, more passage dimension in the birth canal, and also begins to dilate the cervix. In the fetus itself, the weight is going to stretch and distort the layers of the uterus, and the fetus's pituitary releases oxytocin in response to the estrogens that are coming from the placenta. So the fetus releases the oxytocin. It's almost like the fetus is saying, now it's time. All of these contribute to these various factors. As we grow, the myometrium is stretched. Maternal oxytocin is released by the high estrogen levels complementing the fetal oxytocin. And prostaglandin production is a following enzyme, and they will stimulate these cyclical contractions, making them more intense and closer together. This sensitivity that's developing in the myometrium progresses as these hormones climb up and reach a critical stage. And late term, it says here the uterus is 100 times more sensitive to oxytocin than the smooth muscle in a non-pregnant uterus. So labor contractions begin, as I said, mild and far apart. They are an example of positive feedback because the stimulation to a small contraction feeds back to the original nerve pathway, firing it again where it will occur in a shorter duration of separation and at a higher intensity. And this positive feedback continues to amplify the labor contractions. Before labor begins, this positioning is ideal, with the head of the baby downward and the alignment so that the muscles of the uterus can move the baby's head in that direction. Now, the problem in birth is the size of the head. This is the largest and most solid piece of tissue in the fetus, whereas these parts do have bones. The joints are extremely flexible and not very well formed. Most of them have cartilage surfaces, so that this part of the body from the neck down can kind of wiggle back and forth as it approaches this birth canal and basically scoot through. But this big head, these plates in the skull are incompletely formed and kind of can slide over one another, giving a particular shape to the head if the head is large compared to the birth canal opening. So stage one, dilation, this cervix, after long contraction, holding the development of all of this weight up above, because when the mother is standing, the weight is falling directly down on a cervix, and a competent cervix will hold that until the appropriate time for delivery. Dilation occurs as a result of hormonal relaxin, and relaxin causes this to dilate at the same time you're softening the pubic symphysis, giving as much room as possible. Stage two is expulsion at this point. Generally, you can reach in and grab an arm and basically pull the baby directly out. This movement of the baby out will stretch the umbilical cord, and the umbilical cord typically will be cut. But the last stage, the placenta at this point becomes what's called a deciduous tissue. It, in response to these hormonal factors, it separates from the wall of the uterus, leaving the uterus empty and leaving the wall, the accessory tissues detached. Here is the so-called afterbirth, consisting of umbilical and placental tissues. Now, emergence is just the first step. The neonatal period is development, then infancy. During infancy, we talk about infants, we talk about toddlers, we have all kinds of names for the different stages, and we eventually get to that I can run, I can jump, I can talk, I can eat my own food, childhood, leading to adolescence. Adolescence is sexual maturity. You cross over into an adult stage, and then you are a mature individual once you are reproductively competent. These different stages of development are shown here. Infant, a little bit. This infant is pretty much helpless in the face of gravity, whereas this one, you notice, has been able to sit on its diaper upwards. It seems to be holding its head and directing its gaze straight at the camera in response probably to a lot of encouragement. After that, you go through childhood to adolescence and to maturity, and a long period of healthy life in maturity. At the other end of that, maturation leads to aging and the decline into death. So, postnatal life. After you're a fetus, you will depend heavily on the mother's breast milk. Mammary glands are specialized sweat glands that develop slowly over the history of mammals. They basically are a converted sweat that has been modified to produce nutrient-rich food that's appropriate to the baby, containing lots of electrolytes, a lot of nutrients, precursors like amino acids and lipids for membrane and protein development. Milk and our dairy industry definitely shows the value of mammal milk. It's one of our major food groups and is described often as the perfect food for mammals. There's also a lot of lysozymes in milk, so that there is some antibiotic and immune properties that are transferred from the mother to the baby as it nurses. Now, I want to point out what's being transferred are enzymes and chemicals, including antibodies, but not the cells that produce them. So as long as the baby continues to nurse, there is this transferred, natural, acquired immunity, protection against infection. However, as soon as that nursing stops and that transfer stops, when the last antibody molecules wear out, that baby is now susceptible to those things that the antibody was addressing. Now, the baby has the ability to, through having disease or having vaccination, to develop those properties where it can generate the antibiotic in its own body. But that is then a natural immunity, but it takes the infection to where the offspring's body can actually produce the antibodies on its own. Now, the milk production is supported by an enzyme called prolactin. That's what causes the breast to begin milk production. But the letdown reflex is a combination of the hormone oxytocin and the physical stimulation of the external nipple. So the mammary gland is triggered when the infant sucks on the nipple, and it continues to function until that nursing stops. Typically 1 to 2 years is a good estimate for mammal nursing periods, although there have been social influences on mothering and childbirth that have changed that dramatically. I know that in the 1950s, and I'm a little knowledgeable about that. I was born in 47, but I watched my 3 siblings being born from 50 to 56. And my mother was a registered nurse. She used to explain lots of things. And she said at that time that she really believed in nursing her own children, even though the medical community was very strong on formula nursing. Get that baby on the formula. It's scientifically developed. It contains a perfect balance of everything we know the baby needs. So for a while, it was very stylish to select formula feeding as the preferred alternative. Then we found out, we didn't know what we didn't know, that there were other advantages to breast milk from the mother that had been produced in the mammals and in human beings over anywhere to up to millions of years. And so the advantages of natural breastfeeding are now well recognized and well known. Here we see the sequence. Milk is ejected from the stimulus of this tactile response. You'll notice the impulse transmission is from the tactile receptor to the sensory synapse up to the hypothalamus. The hypothalamus's particular role here, because there are 2 very prominent enzymes produced in the hypothalamus. One is ADH, which we've talked about affecting water balance, and it's primarily through action in the kidney. But oxytocin is the other. Oxytocin is produced by the hypothalamus and stored here in the posterior lobe of the pituitary. Under this stimulation, oxytocin is released, and its primary effect is what's called milk letdown. And so the milk flow is allowed and flows to the nursing infant. Infancy and childhood, as stages of development, involve the activation of all the hormones we've talked about from the beginning. Growth hormone is a general stimulating hormone for cell division and differentiation and for metabolism in general. It holds it at a high rate and growth is rapid. Artificial experiments where we take growth hormone and apply them artificially through supplementing feed shows that it can have a stimulating effect. Basically, experiments with proper controls have been conducted where growth hormone is added to the feed of mice and withheld from the feed of the control group. And you basically get mice, genetically the same population, but they grow to the size of rats. Adrenal gland and its steroids and thyroid hormones are also stimulating. Adrenal gland we saw with fight or flight reaction with an overall stimulating effect for a local emergency. Thyroid in general, probably someone who has a thyroid condition or low thyroid and they feel tired, they feel fatigued all the time. Thyroid hormones have this general stimulating effect. Growth does not occur uniformly. It occurs in spurts. So the development of the egg, fertilized egg, to the infant involves extremely rapid cell division. That first year of growth, it's extremely rapid. Although more pounds may be put on in another year, the weight doubling does not occur again. There are growth spurts as associated with puberty, especially in males that shoot upward and their shoulders get broader and 3 shoe sizes in 1 year. Proportions change, so the bodily changes in a woman's body are as dramatic as the breasts develop and the hip girdle expands laterally to produce the typical differences in shape between male and female. Puberty is where we begin adolescence. And it continues until your growth is completed. Now, since we're so fixed on height, we typically think we grow until we're 18 or 19. And in fact, height often is completed by 19 or 20, but bodily growth overall continues considerably beyond that. So a better estimate of the end of adolescence would be 25 to 28 years old. Through that middle period, the end of adolescence through maturity, you have a situation where the rate of cell division and the rate of cell loss are pretty much balanced up and down. Now, injury or disease can throw it out of balance, but basically the body, when it recovers, it goes through decades of time where cell division and cell loss are basically balanced. This is that long-term healthy middle life. At some point, however, the rate of cell loss, whether it accelerates and the cell division diminishes, or whether the rate of cell loss actually accelerates due to some genetic or environmental condition, you reach a stage called senescence that leads to a recognizable change in your capacities or aging. The capacities of an individual basically diminish. Osteoporosis lightens the bones. You lose muscle mass. Your brain shrinks. All of these changes that are typical. Pigment production diminishes so that your skin tone may change. Your hair loses its pigment and turns gray. In some cases, it falls out of the follicles. The epidermis thins. It becomes more subject to injury and so forth and so on. We have basically looked at aging in every organ system up until now. This affects the homeostatic mechanisms. One systems changes, impacting the other systems that depend upon it. Along with this, we see a decline in the sex hormones. So testosterone and estrogens both diminish as you approach the end of your fertility. On all organ systems, you have a number of kind of characteristic functional changes, and you'll recognize this when they're in list forms. But it might be helpful to do a bit of a summary, because this is summarizing things we've talked about from the beginning of A&P1. In the skin, it becomes thinner, less elastic. So it becomes basically more prone to tearing or cutting. That looser skin, that thinner skin, also sags with gravity, producing wrinkling and other outward effects. Bone deposition changes relative to bone consumption, and the bones become not smaller, but they become weaker. Osteoporosis is like the bone is no longer solid, hydroxyapatite, and well mineralized. It develops little pores or little bubbles, and it becomes progressively lighter. You can actually see this over a period of years on x-rays as the outline of the bone becomes more and more faint, as if the bone is disappearing. You also see it losing weight and more prone to breaking if you fall. Now, combining that with what's happening with joints, joints are losing their range of motion as connective tissue diminishes and becomes stiffer. And also, a lot of the joints become worn to the point where they're no longer as well lubricated, or the ends are no longer covered with as much cartilage, and pain signals begin to, not only do you lose range of motion, but our typical reaction throughout life when we encounter pain is to not move it. And so as a result, we get in this downward spiral. Pain causes us to move less. Moving less causes the joint to get stiffer and send more pain, and so forth. Muscular strength and ability to stretch and relax is reduced, and the muscle mass of the body goes down. That loss of strength has a number of far-reaching effects. So, for example, getting down on the floor and getting back up becomes very difficult because of the limit of muscular strength and of elasticity. In the brain, we see a loss of coordination between the cerebellum and all of its somatic motor system activators. Memories from the cerebrum, intellectual function, the ability to secure and develop new memories and turn them into secondary and tertiary memories is impaired with aging. Circulating hormones, almost all the glands are diminished in their production. So starting with the general senses, things like the ability to sweat is affected. Things like the ability to produce skin oil and pigment is affected and reduced. Within the endocrine glands, all diminish in their production, including the sexual hormones. Cardiovascular problems are a main part of the difficulty of aging. The heart's function is affected in concert with the vascular system, and peripheral blood flow is reduced. It can affect a variety of organs, and so pain or loss of sensitivity in extremities, issues like neuropathy, blood flow through the blood vessels in the brain, the blood flow to the extremities like hands and feet can cause problems. The immune system and the ability to generate immunity in the face of new challenges, new antigens entering the body, are reduced as well. So in reflecting on that single comment, it fits in perfectly with what we're hearing about the coronavirus being disproportionately effective in leading to serious illness or death in older people. I now have encountered enough comments on the news in the last couple of months about extended care facilities and facilities where all elderly people are in living groups to understand that decrease or desensitization of the immune system is having a big impact now. The lungs, less elastic. So you no longer get as much oxygen with each breath. So another reflection on coronavirus where respiratory influences are linked with death. Muscle tone along the digestive tract, so peristalsis, and muscle massage or propulsion is decreased and digestion is impaired. In the urinary system, the same thing. The loss of muscle capacity means the movement of urine to the bladder is impacted. It also means inside the cardiovascular effects are affecting blood flow to the kidneys and the amount of urea that can be removed. That first step of filtration is basically supported by blood flow and blood pressure. The reproductive system eventually becomes active. In the female, menopause is simply when the cycle of ovulation stops for good. In males, it's called a male climacteric, and it's more extended, less predictable in terms of age. However, it is there is a reduction in sperm production, a reduction in both the number of sperm that are produced and stored, and in their ultimate activity. So if you look at sperm longevity, how long do they swim? As you look at them through the microscope, the sperm exhibit less endurance. They exhibit more malformed sperm, and that is a process that continues with increased age. So Development now has brought us all the way to the diminishment of systems, and we've summarized how aging or a condition like senility affects each of the organ systems. Our final major topic is that of inheritance. In this case, we go back to the 1860s and the publications of an Austrian monk named Gregor Mendel. There were many conflicting ideas about how inheritance was affected, what these inheritance particles might be. It was clear there was a relationship between parents and offspring by the consistency of reproduction. If you study salmon, you collect their eggs and mix it with their sperm, what hatches out is a fry that grows into a salmon. So there's a consistency of inheritance. The expression is like begets like. So human beings produce more human beings, not other species. The characteristics that seem very important in humans are the chromosomes within the nucleus of the cell. They exist with a characteristic numbers. In humans, it's 46 chromosomes. But further study and really quite recent techniques, those from the 1970s and 80s, now perfected, that allow us to study the chromosome structure of each chromosome, has revealed and confirmed that those 46 chromosomes exist as two complementary sets of chromosomes. So the process of sexual reproduction, which always contains meiosis to produce gametes and the fusion of gametes to produce fertilization. Meiosis is a process that takes one of the father's cells and breaks it down into a half set. That means one, if you look at the number one pair of chromosomes, one of those is going to go into each gamete. If you look at the number two pair, one of those is going to go into each gamete. So there's going to be a 1, a 2, a 3, all the way up through a 22. And because the final pair in males is an X and a Y, either an X or a Y. So it's not just 23 chromosomes out of the 46. It's an exact half set. Likewise, meiosis in the female is going to produce an exact half set of her chromosomes, the only difference being, since she has two X chromosomes, an egg will always carry an X. So that means when a sperm joins an egg, you'll be carrying one number one chromosome into an egg with one number one chromosome, and you will have a pair of number ones. You'll notice one from the mother and one from the father. One member of each pair is contributed by the sperm and the other by the ovum. This is the only way a reduction division can work. If you reduce the chromosome number in a very exact way, and then combine it with another cell where it's been similarly reduced to restore that consistent number. Let me just make this comment about chromosomes. There are groups where chromosome number and chromosome type are very variable. Plants are notoriously variable in their chromosome number. In fact, changes of a chromosome number of 1 or 2 chromosomes is often associated with a speciation event. Plants can also multiply whole sets. So if a plant has, as one plant I studied, had 24 chromosomes, 2 sets of 12, and I discovered a plant that was very similar, it looked like the same species, that had 36. And it was produced that way when probably when a pollen grain with 12 chromosomes contacted a megaspore that was unreduced. It had 24. So those 36 chromosomes did not produce a balanced set. It turned out that in plants, because of their propensity for vegetative reproduction, that plant with 36 chromosomes produced quite viable and robust individuals that grew and flowered and reproduced very avidly vegetatively. But when you looked at their pollen, the pollen was over 99% sterile. When you looked at the megaspores did not pair regularly and produce regular megaspores. So the main effect on multiplying chromosome sets or polyploidy in plants is to make them sexually sterile. Now, that picture in plants is very different than the picture in animals. We do not find a lot of chromosome variation in live-born human infants. There are only a few changes to chromosome number that are tolerated, and those typically bring an associated level of recognizable disability with them. So you've got your 46s. The highest tolerated variation in human chromosome number is an extra X or Y, or one less X or Y. So we do have numbers of 45 and 47 associated with the sex chromosomes. In fact, the most common chromosomal difference is having 3 number 21s called Down's syndrome. These individuals all have a recognizable level of disability, and they also have lowered sexual fertility. There are, in addition to XX and XY normal complements, there are individuals who live normal lives and have XO, so a total number of chromosomes 45. They carry one X chromosome and present as females. Likewise, there are XXX individuals who have 47 and present as females. There are some individuals that are XYY, and XYY individuals present as males and have 47 chromosomes. Other than the sex chromosomes, there are very few chromosome changes in humans that are tolerated, and this is a kind of a general rule for animals. Here is an arrangement, a very specialized visualization of the chromosomes from one cell of one individual. When we do this, we have differentially stained these chromosomes, typically with a protein stain after a base treatment. And the characteristic banding pattern allows you to, because of the correspondence of banding, match up the chromosome pairs. So you can see the size and the primary constriction of these 4 chromosomes are similar enough that if they were uniformly stained, you wouldn't be able to tell who was who. But there's only 2 that have this big purple region on the end. These chromosomes do not carry it. There is only 1 that have this big green and large and medium-sized green band here and here. These have more reduced green. So it's not hard to match up these as homologous pairs. And as you go down through this, you can pair these chromosomes showing a normal complement. When you present the chromosomes in this way, as pairs and arranged in order of length, we call this a karyotype. Here's the word right here, karyotype. And this can reveal changes in chromosome number. Another thing I can tell from looking at this karyotype is these chromosomes are male, because we go 1 through 22, down to the smallest. But it is traditional to put the X chromosome, which is really quite large. It's about the same size as the 7 or 8. And the Y chromosome right next to each other at the end. So this XY pair shows this is a male. An individual who is XXY would have an extra X chromosome here and a total of 47. An individual who is a Down's syndrome individual would have 3 number 21s right here, and the banding pattern would clearly show that that chromosome is a number 21. Patterns of inheritance involve chromosomes? Because we know that genes are located on the DNA strand that forms the core of the chromosome. Now, there are two different patterns depending on whether the chromosome has a pair. If it does, like if this is the number 1 from the mother and this is the number 1 from the father, we call that a homologous pair. If there's a gene for lactase production here, then this number 1 has a gene for lactase production right there, and those are homologous alleles. But 2 copies are determining the trait that is inherited. Simple inheritance is the simplest factor when there is 1 gene, 1 location, and 2 alleles. About 80% of your genotype falls into this category. There is a difference when you're talking about genes that are carried on the X chromosome or the Y chromosome because they do not have a homologous pair. A gene located on the X chromosome can be carried in a male or a female. In the female, it would just resemble this autosomal inheritance because 2 X chromosomes are present. But in the male, this small Y chromosome does not carry all the genes that are carried on the X. So this is sometimes called sex linkage or X linkage, more appropriately, because the majority of traits that present this way are genes that are located on the X chromosome. Now, I might mention, with respect to sex linkage, there's another phenomenon for the few genes that are carried here on the Y. But the inheritance is very simple. Y chromosome inheritance involves every son that is born because the presence of the Y chromosome makes that individual a boy. So if I have a son, it's because my Y chromosome has been passed. So it is from father to son, grandfather to son to grandfather to father to son, right down the male line. Let's take a look at a couple examples of this inheritance. Major patterns of inheritance are going to be understood more completely the simpler the expression is. So, for example, if it is a single gene with 2 alleles that you see here, one is dominant to the other, meaning if you have 2 reds, you get the trait of red. If you get 2 yellows, that's the trait of yellow. But in a mixture, you get the trait of red. That would be a simple dominant or a strict dominant inheritance pattern. One allele completely covers up the presence of the other one. Dominant traits like normal skin pigmentation, freckles are dominant. Nearsightedness and farsightedness are both dominant traits. Astigmatism in the eyes, free earlobes, tongue rolling as a behavior. Rh factor, the Rh plus is dominant to Rh negative. Type A or type B blood are dominant to type O. Huntington's disease is a disease that results from a dominant allele. If you have only 1 copy, you get Huntington's disease. The complementary allele, always you have to think of what is the complementary trait. So if normal pigmentation is dominant, albino pigmentation is recessive. That means to be an albino, you have to have little a, or if yellow is recessive, yellow, yellow. Now, that means that you show the trait if you're homozygous, yellow, yellow. If you are red, yellow, you do not. You show normal pigmentation, but you carry the allele for albinism. Freckles is dominant to the absence of freckles. Nearsightedness and farsightedness are both dominant to normal vision. Here's free earlobes and attached earlobes tongue rolling and inability, so forth. A recessive trait, cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and phenylketonuria. All four of those disease factors are recessive, meaning you have to have the double recessive genotype to express them, but you may in fact be a carrier without expressing the allele. Codominance. That's where the presence of each allele of the pair is expressed. So the most obvious example that we have available is type AB blood. So AO is type A, BO is type B. But AB is type AB, the presence of both, and the antigen and antibody factors that are present in the blood will be affected by this genotype. Codominance. The different alleles contribute to the structure of albumins, those blood proteins, and also to transferrins, transport proteins. Incomplete dominance is where there's an intermediate trait in the phenotype. So hemoglobin A is one thing that shows intermediate inheritance. Hemoglobin S, sickle cell anemia, shows an intermediate inheritance. If you have only one sickle cell allele, you do not, you are viable, you can be alive. The heterozygote, though, has a reduced viability because of the impairment of oxygen transport due to the sickle cell hemoglobin. Now, I'm going to stop the recording right there because we do have next week to complete this pattern. So let me pull things to a stop. I will take a moment to check attendance.